Okay, so hi everyone and welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Professor Itamar Kahn. Dr. Kahn received the Bachelor of Science in Mathematics and Computer Science from Ben-Gurion University and received a PhD in Neuroscience from MIT. He did his postdoc training at Harvard University. Dr. Kahn has been a faculty at the Technion Israel Institute of Technology at the Faculty of Medicine between 2010 and 2021, and recently joined Columbia University's Zuckerman Institute. He received various awards and honors, including the MIT Angus MacDonald Award for Excellence in Undergraduate Teaching, the American Federation for Aging Research New Investigator Award, and the European Union Marie Sklodowska-Curie International Reintegration Grant. I would just like to remind the audience to please address any question you have using the Q&A box or raise your hand. Professor Kahn, thank you very much for coming here today. The virtual stage is all yours.
Thank you for having me. Can you see my screen and can you see me?
Yes.
Can you see me? Excellent. So first, before I'll go ahead and tell you about the topic of today, I would like to first acknowledge the many collaborators and founders. This work has been done at the Technion, and now we are transitioning our lab to Columbia, and we'll continue this work over here. And I'll mention the team members, the lab students, and the postdocs that has done the work during the talk. So thanks for having me. It's actually an exceptional pleasure for me to be at Martinos back. The origins of my education came from Martinos. I'm going to present today some work that I've done under the supervision of Randy Buckner and Chris Moore, both members or prior members of Martinos. But even my PhD education came at the Martinos. I was working with Anthony Wagner back when he was at MIT, and all of our scanning was done at MGH. And even before that, before I was even in the US, I worked with Rafi Malach, who brought the fMRI or bold imaging to Israel after spending his sabbatical with Roger Tutel at MGH. So really all of my education came from Martinos, and I was educated there, and kind of my fMRI became my passion, I think, somewhat because of Martinos. So I would like to tell you a little bit about the work that we've done over the past few years, establishing a platform for awake imaging of mice. And to motivate this presentation, I brought this photograph. So what you're seeing here is a photograph by Sam Raleigh, submitted to the Natural History Museum Wildlife Photograph of the Year competition a couple of years ago. Sam discovered that the best way to photograph the mice inhabiting London's underground was to lie on the platform and wait. He only saw them fight over scraps of food dropped by passengers a few times. And what you see here in this instance is one of these fights. The fight lasted a split second before one of the mice grabbed the crumb, and they both went their separate ways. I found this photograph visually arresting, and I found it to be a good way to introduce my own interest in human and animal behavior. It shows these two mice actually showing very nice dexterity in their digits. So you can see them standing on their hind paws, both of them trying to grab this piece of crumb using their kind of digits. And also showing the interaction between these two mice. So if one of them would have been a rat, obviously they would not try to fight, the mouse would not try to fight the rat over this piece of food. So they are conspecific, so the social interaction aspect. And also it's an excellent demonstration of goal-directed behavior. The animal being hungry, identifying that it could forage for food in this environment, but also acknowledging there's competition, the need to kind of fight for it, and then, you know, when you lost, and kind of you move on to find another piece of food. John Krakower's perspective in Neuron on the role of studying behavior and the need to correct for bias in the field to focus on circuit level analysis is capturing well the motivation of the platform that we developed for behaving mouse fMRI. So this has been something that was influential on me for a long time, and John really articulated well this need in neuroscience research. And I'm actually citing from something that he cited in this piece that he wrote from William Bechtel. A reductionist treatment of the components must be combined with investigation of how the total mechanism is organized and how it behaves when embedded in an environment. So really, this to me captures our need to understand, if we want to understand behavior, we want to understand the brain, we need to have a good whole brain view of what we're looking at. And that points out to one of the main challenges when doing animal work, is that the approaches that are available in animals are actually not so effective in giving us whole brain coverage. So now there are, there are a couple of methods that are emerging as ones that give you a wider field of view, but I would like to argue that fMRI is actually a great approach to give whole brain view even better than the others. This is just one example from John Crocker's perspective, kind of highlighting that whole brain imaging methods are going to be especially useful in scenarios where multiple known or unknown circuits may contribute to a single behavior. So we can look at the whole brain, can look at different patterns of activity and see how these kind of mechanisms, these neural substrates contribute to that specific behavior. Now thinking again about animal work where we have a lot of tools at our hand, we can use tools like optogenetics or chemogenetics to try and causally manipulate behavior. And this is the general motivation for doing whole brain imaging with fMRI in mice, as well as looking at behavior. So not just being able to look at, say, resting state signals, which we have done, but also looking at the animal when it engaged in behavior. So for today's talk, we just went through the quick introduction, and the talk will be in two parts. The first part is actually going to focus on the mouse fMRI response, looking at the mouse hemodynamic response and awake imaging. This is work that I've done during my postdoc with Randy Buckner and Chris Moore, and that served as a motivation for developing the platform for awake behaving mouse fMRI, and that will encompass the second part of the talk. I'll talk about, I'll present to you different platforms that we built for looking at locomotion, reach, and leak, only briefly showing the feasibility of these platforms. I'm not going to go into too much detail. And then for olfaction, I'll show some more brain imaging results, looking at learning in the brain as a function of time, and show a paradigm that really, in my mind, highlights the benefit of doing whole brain fMRI in the mouse. And at the end, I'll describe a few future directions that we are kind of now engaged in at the Zuckerman Institute. I'm actually going to skip a lot of work that we've done over the years in my lab, looking at cross-species comparative brain imaging, doing brain mapping in health and disease. We've done all of these with resting state, again in the awake mouse, but in passive wakefulness and not during behavior. So I'm not going to discuss any of these results today. So just to give those of you who haven't thought or haven't encountered brain imaging of the mouse, what it encompasses, really the main challenge of doing whole brain imaging in the mouse using fMRI is the size of the brain. So it's actually not the brain of a mouse, it's an elephant shrew, but it's the size and scale of a mouse brain. And I like this depiction because it really gives you the scale of smallness, of how small this mouse brain or this elephant shrew brain is relative to the human brain, and the fact that really we need high resolution, high field imaging in order to get good brain coverage. So this will be 3 mm cubic voxels So obviously that cannot be used to study the brain of a mouse. Even if you go to higher field in a kind of human, it's still not sufficient. This is 150 microns, and this is 100 micron coverage. So we are actually able to get to 100 micron coverage. Most of what I'll show you will be either 150 or 200 micron in-plane resolution and about 400 or 500 micron slice thickness. That gives us still a very good resolution of imaging, even including some basic laminar level resolution, but definitely excellent coverage of hippocampus, striatum, thalamus, brainstem, cerebellum. So great resolution. We are all the data that I'm going to show today is all done with spin echoplanar imaging. We did that in order to kind of get more sensitivity to hemodynamic responses from the arterioles and capillaries, trying to avoid as much as possible the large draining veins. There are still contributions when you do T2 imaging for functional MRI because of the T2 prime. There are now methods actually at the Martinos developed to try and suppress these contributions. So definitely we are going to be users of such developments. So everything is spin echo EPI. So the first set of studies that I'm going to present are all studies that we've done in order to convince ourselves that we can do functional MRI in the mouse, we can do it in the awake mouse, we can map networks, we can estimate what is the hemodynamic response function like in the mouse, and that these things are reliable across multiple animals. So this was the main motivation. Working at Martinos, working with Randy Buckner, one of the first ideas that actually Randy came up with was to try and replicate the work that he's done in the 90s with Anders Dale, looking at the temporal linearity of the hemodynamic response. And we were well positioned to actually do that in a very causal manner, to activate cells in the brain, to compare activation of such cells to a sensory drive, and look at temporal linearity. That's the result that I'm going to show now. What you're seeing here is an animal transgenically expressing channel rhodopsin 2, so we can use light to optically drive activity in cells, mostly in layer 5 of the cortex, throughout the cortex, a little bit also in layer 2, 3. On the left, you see the expression of this opsin, and in the blue bar shows you where the light guide was positioned. We did electrophysiology to confirm that we have a precise control of these cells, that they are indeed producing action potentials. And then we did two conditions. One of them is an optogenetic drive. The other one is sensory drive. We used an interleaved design where we have some runs where we are showing a block design, so 15 seconds on off of either optogenetic drive to this location or sensory drive, which is whisker deflection to the mouse whiskers. And then interleaved between these runs, we had the target runs, which were our critical test. For this, what we've done is we wanted to compare 1 second optogenetic or sensory drive activation versus twice 1 second separated by 2 seconds. So here you can see on the left the optogenetic drive. So this is the location of responses that we got from the block designs. We identified these voxels, and then we extracted the time courses from this region of interest, and we plotted them for the 1 trial condition in blue, for the twice 1 trial condition in red. And here you see in the dashed line, you see the linear summation, which is the blue line shifted by 3 seconds and added to itself. And what you can see here is a pretty good match between the 1 second when it's shifted and added to itself and the 2 trials in red. On the right, you can see the sensory drive for whisker deflection, showing exactly the same type of response. You can see that this response is a little bit longer, but still it behaves in a linear manner. Here what I'm showing you is for each of the individual animals that we've ran, a triplet of time courses. In blue, it will be the 1 trial, in red again, the 2 trial, and in dashed line, instead of doing the linear summation, now I switched it to be linear subtraction, just to allow you to appreciate for each individual animal that we see, if we subtract the blue from the red, the 1 trial from the 2 trial, we now get a dashed response that looks like the 1 trial shifted by 3 seconds in time. And again, this response holds for the sensory drive as well. Here on the left, you see 6 animals. Here on the right, you see 2 animals. Again, all of the animals show this patterned response, all of them showing temporal linearity. So that was a fantastic result. We got very excited by it, but we wanted to push it further. So one of the questions that remains from temporal linearity is when are these conditions fail, and whether for sensory drive has been shown in humans where temporal linearity does not hold or does not hold to a first approximation, whether it would hold for the optogenetic drive, and what would be the result for the sensory drive. And to test that, we compared 5 trials versus 6 trials. So now we took it to the 5 trials versus 6 trials. We did it on 2 animals. And what you can see here, so now you can see that this is almost like a block design. So you have multiple events showing up. The entire response is a prolonged response, but if you subtract the blue response from the red response, you still get this punctate single trial response, similar to the one that we got for the 1 versus 2. And you can see that actually for the sensory drive, we get some saturation in the response. And this result is important because it really disconnects the fact that we are not getting this type of kind of very clean linearity in the sensory drive from the possibility that the neurovascular coupling is the one that is saturated or having a ceiling effect. So we really see here that still with optogenetic drive, we get temporal linearity. The next set of studies that we conducted were aimed to try to look at the network or brain-wide response, also to move from the anesthetized to awake animal. So this first study that I described was all done in the anesthetized prep. And what we've done here is we used 5 individual animals instead of the transgenic line, the Taiwan CHR2 transgenic line. Now we use the lentivirus to express channel rhodopsin in a primary somatosensory cortex, so very similar to the location that we had before. We drove responses in a block design manner, so 15 seconds on off in these animals, and we computed the whole brain map. You can see here the median correlation map to the block design that we conducted. It looks to be something that can be the network of regions that are connected to primary somatosensory cortex, but we wanted to validate that indeed that's the case on an animal by animal basis. We also wanted to evaluate the consistency or the kind of how much it replicates across different animals, how much it fits kind of the understanding that we have through Atlas of the brain. And in order to do that, we used the non-supervised clustering algorithm to try and cluster together these responses within and across animals and to match them to the Atlas. And that's what we are plotting here. Here you can see the contribution of the 5 different animals that were participated in this analysis, or included in this analysis. And you can see here the kind of clusters as were determined for primary somatosensory cortex, secondary somatosensory cortex, primary motor, and contralateral. You can also see the caudate putamen here response, and it seems to be a pretty good network response across these different animals. We evaluated this in a number of ways, And indeed, we're convinced that to a reasonable degree, this is a good response that shows up across the different animals. So a brain-wide response for this network. What we then wanted to do is to compare what would be this response if the animals were anesthetized. So in the same animals, we subsequently anesthetized them and computed what would be for these regions of interest that were identified using this unsupervised clustering algorithm, the percent signal change. So you can see about 3% signal change for these 5 regions in the awake state. And you can see a reduction, but still a reliable response in the anesthetized case. So we can really use this optogenetic drive to identify the network in the anesthetized case. However, we do see, if we do this normalized cross correlation, we do see a differential kind of reduction between the cortical and subcortical regions, in this case, just a clouded putamen here. But also, if we actually would use the data set of the anesthetized animals to do this unsupervised clustering algorithm, this is what we are getting. So now, instead of having a reliable way to identify responses across the different animals, what you can see here is that all of the subcortical responses are gone. We are still left with the cortical responses, but even those are more spread out, seem to be more diffuse relative to the awake state. Now that was a good convincer for me, now kind of on the verge of embarking and opening my own lab, that really we should kind of make a significant effort, if not for having the animal behaving in the MRI, which is impossible without it being awake, also for just network mapping of these animals, the awake state will be preferential. So before moving to talk about awake animals, I just wanted to quickly note that we did additional studies to look at the high resolution fMRI. This is again the study that was done with Mittal Desai and under the supervision of Chris Moore and Randy Buckner, looking at the kind of laminar responses in these animals. So this now we are doing in high resolution MRI for what was possible for us at the time, 100 microns in plane. You can actually see the profile here of responses. You can see that the maximal response is at around 800 microns in depth, so equivalent to layer 5. If we are looking at electrophysiology, both multi-unit activity and local field potentials, so you can see that the multi-unit activity is maximal around the region where we are seeing maximal responses with the fMRI, while local field potentials are more superficial. We've done another study in order to look at these responses more in depth, doing more electrophysiology, but also looking at different drives of optogenetic activity. Here we compared different rates of frequency of stimulation, so between 8 Hertz and 80 Hertz, either in Poisson distribution or a periodic distribution, measuring also the single unit activity, local field potentials, looking at kind of now at instead of temporal linearity on a rate linearity. So if we increase the rate of activity, what is the expected change in the bulk response, and what would be the change in reality in the bulk response. So you can see here that there is a kind of a monotonous increase of activity. You can also see and appreciate that Poisson drive is a better driver for this activity relative to the periodic drive. To remind you, these are pyramidal cells of layer 5, so this is to be expected. And what happens when we are doing these different measurements now with kind of laminar probes, comparing them to the single unit activity and now with multi-unit activity. We also had another measurement of the direct current LFP, so a different way of measuring LFP, avoiding kind of some of the pitfalls of kind of AC modulated LFP, which we think was underlying the kind of the reason of failure to predict bulk responses with LFP. And you can see here that the maximal response for multi-unit or single-unit activity is indeed at around layer 5, so similar to what I've shown you before. And you can see that the local field potential responses are more superficial. So again, matching what we understand about the physiology. Now, if we're trying to estimate the transformation of electrophysiology to bold responses, we used either first or second polynomial order functions. We looked either at the early or late components of a 15-second response interval, which is what we had. You can appreciate that first, second order is outperforming first order in all conditions, and that early and late for the second order are good matches. And finally, that single unit and the direct current LFP and multi-unit activity are the best predictors for the bold response, again, for pyramidal cell activity that we are driving exogenously, so with optogenetics. So really kind of highlighting that if you know the electrophysiology, know what are the participation of cell types, this method allows you to estimate what would be the bold response for that in that region. So that kind of set the stage for us and for me personally to use this method to look at fMRI and within the behaving animal. And before showing you the platforms that we developed, I would like to just briefly note that there's been a lot of work developing methods for high resolution fMRI. One example is from Xin Yu, now at MGH, doing beautiful work on a number of papers. Here is just one of my favorites, line scanning fMRI, which gives us a very high temporal resolution with a good spatial resolution when you're agreeing to be limited to a very small portion of the brain, but then allowing you to resolve temporal onsets of different cells. Again, here it's also a good demonstration of the use of optogenetics. This is from the lab of Sunji Kim, now in South Korea, showing the benefits of ultra high field imaging to look at network connectivity and directionality in the mouse's brain. Again, using optogenetics and also kind of a sensory drive to look at these dynamics. And to me, this is all binds together to our own effort to do awake behaving imaging. So to leverage these technologies in combination with the platform that we developed. Okay, so now let me switch to the second part of the talk and show you a couple of examples of the different behavioral modalities that we developed and also some brain results on what we can study with this platform. So the basic kind of platform that we built was actually something that Mitul and I developed back at 10 years ago, or actually more than 10 years ago, almost 12 years ago, to have a head-posted animal. In Israel at the Technion, we took it to the next level by having everything 3D printed, so we can kind of print the different parts. Here you can see the cradle where we will position the single element coil in order to measure responses in the brain, the head post that we're going to fix to the animal's brain, and then the bridge that we're going to hold the animal in head fixed position. This is how it looks with the animal in the cradle. This is a top view, and this is a side view. You can see here the animal's head. We remove the skin and essentially attach dental cement to the scalp in order to have the head post attached, and then we can put it on the bridge. And here you can see a single element 20 millimeter coil, which we can use for whole brain coverage. Just to kind of show you that even in the first trials, Eyal, who kind of pioneered all of that in my lab in Israel, could already evoke behavior. Now you can see a mouse in this cradle where we are putting a water tube right in front of it and give it water drops, and the animal immediately kind of uses its paw to pick up the water and leak it. So even in this very simple apparatus, you can evoke a rich behavior. And that convinces that really we should invest a lot of effort in having a well-controlled behavioral platforms, both for motor and sensory drives, and I'll show you. So the first apparatus that we built was for locomotion. Here I'm just going to show you the apparatus itself. This is the cradle that Guy Yona built. It's the same cradle, but now with the mouse standing on a treadmill, allowing it to move freely. We have here an encoder that tells us when the mouse is standing or walking. These parts are actually from Lego, both the track and the wheels here. And we can also now use a camera, a video camera that is MR compatible to do imaging of the behavior with videography and use DeepLab or similar tools to actually classify the behavior. The lines that you see here are the artifacts from the gradients running. These actually average out if you want to do any type of analysis with DLC, so they are not huge artifacts that disrupt. And you can actually appreciate that you can see here the nose of the animal moving, the whiskers moving, so not individual whiskers, but the overall kind of facial face of the animal. And we are now in the process of using that to also do facial expression recognition of the animal in different conditions. So that's for a locomotion. Another apparatus that we built was for reach. Here I'll show you a little bit of brain data just to convince you that you can do this whole brain imaging from the cerebellum all the way to the olfactory bulb. Here the animal actually gets a light that tells it that the water drop is going to appear, so a classical conditioning type paradigm. And this is actually outside of the scanner, just showing the period where we are training the animal to do this outside of the scanner, to get used to getting water outside of the scanner. You can do the animal doing this leaking. And now you can see that the animal inside the MRI. So now this view, you see the animal from the front. You see two dots that are located on the animal's paws. And actually there is a side mirror here, so a mirror that is 45 degrees angled that can show us the paws of the animal from two directions. So we can triangulate it in space. You can see the tip of the nose of the animal, and you can see the paw moving, and you can actually even see the individual digits of the animal. So we can kind of track the animal's movement in space. This is just showing that we can kind of use that to identify responses across the entire brain. You can use it to look at the different movements of the animal, how it does the reach, when it learns, when it becomes more proficient, and the overall process of motor control. You can combine it with optogenetic suppression or activation in order to see disruption in the components of the network. So really a very extensive platform to study motor control and motor behaviors. Another kind of improvement that we've done on that was to actually look at the leaking behavior. Now you can see the entire apparatus. You can see the mouse. You can see that there's a water spout in front of it. There's a mirror on the side, and you can see the camera from the front. Here is what the camera shows, and I can show you a movie of that behavior. Start. You can actually see the tongue moving in and out. We can actually track it. This is very interesting because when the animal is learning this association, we can also look at an animal that is hesitating. For example, when it understands that maybe one cue is predictive of a reward and another is not. So not only the animal kind of collecting the water, so you can actually see the water spout kind of moving, and we can detect that with the pressure sensor. We can actually now look at the tongue itself and look for kind of the animal hesitating if it's unsure about its responses. And again, you can see also the face of the animal. So in this paradigm, which I'll just briefly show you, we had three different classes of conditions, a cue that is predictive of reward, no cue and reward showing up surprisingly, and then this is with the blue light. This is no light, and then a reward appearing, and then a yellow light that is not predictive of reward. How does the behavior looks like? So on the y-axis, you can see the rate of leaking. On the x-axis, you can see the time from the appearance of the reward itself and backwards to the queue. So this is the shaded area. And you can see it as a function of learning. So we run this day after day, so you can have the same animal in the scanner day after day because we're not kind of anesthetizing the animal. So you can really look at the process of behavior and see kind of the progression from a naive animal all the way to a proficient animal. Just quickly, the coding of this is leak plus reward, so LPR, and then different magnitudes of reward. So a lot of reward or little reward, and to look at the different kind of responses to the magnitude of reward. And you can see that for the LPR, the animal is developing an anticipatory leaking response, so it knows that there's going to be a response later. We don't have anticipatory when the queue obviously appears surprisingly, and it does develop an anticipatory response for the light-only condition. It's actually at the end of this, by kind of day 15, they start to show suppression of these kind of anticipatory responses. The reason why we see them, and I'm not showing the data here, is because the animal is actually water deprived. In order for us to have it in a head fixed position, we do water deprivation for it to have kind of interest in leaking water from the spout. And so it has vigor and it will still leak even for this light, but we do have an indication of learning. Looking at the brain, I'll just show you one set of results from the nucleus accumbens, showing kind of again the progression of learning from light to dark. This is the progression through the different days, and the animal kind of gets a robust response by the kind of about third through this process. We can look at the different responses in the nucleus accumbens. So now you can actually see that there's a prolonged response with what seems to be multiple kind of epochs of responses. So we don't have electrophysiology for that, but that's something that we are interested in kind of now acquiring to try and understand the different components of responses. And here, just as an example, kind of the reward prediction error signals that we are expecting. So seeing that this initial response is showing gradation for the different types of conditions with the kind of the magnitude of reward for these different trials going down from a surprising high magnitude to the light only with no difference post-learning, and almost the opposite for these lateral responses. So again, dissociating components along this response. Okay, so for the next 10 minutes, I would like to show you the last paradigm that we developed that afforded us the best kind of monitoring of the behavior of the animal, and that's a GoNoGo auto discrimination. I'll quickly show the paradigm itself. We presented one of two odorants. The odorants that we used were ethyl acetate or quinine, and we counterbalanced them across animals. One of them was assigned as the Go odorant, and the other one is the No-Go odorant. The animal had response window in which to respond in a Go-No-Go fashion to the odorants. If it would leak for the Go odorant, it would get a water reward. If it didn't leak, nothing happened. If it would leak for the No-Go odor, it would be a false alarm and it will not get a water reward. And again, a no leak is a correct rejection. Again, not getting a water reward. So here you have the entire paradigm. You can see that there's no overt punishment for the animal leaking for the false alarms, but it should be noted that even the absence of a reward is a kind of a motivator for the animal to avoid these type of responses. So while a strategy to leak air all the time could be an effective strategy in this paradigm, you'll soon see that the behavior itself is not consistent with that. So what was the experimental protocol? This is actually very similar to the other platforms. How do we get the animals to do behavior in the MRI. So we do the head post surgery where we implant the head post itself. If one would be interested in doing optogenetic drive or putting a window for optical imaging or for two-photon microscopy or things like that, that will be the stage that one would do that. Then we put the animal under a regime of water deprivation, gradually decreasing the amount of water that it gets and not letting the animal have water ad libitum. We are doing a pre-training stage where we teach the animal to actually leak to get water from the water spout, so bringing it to the lab every day on the same time during its diurnal portion of the light-dark cycle, and then teaching it to get water from the water spout. One day acclimation in the MRI with the MRI apparatus, again with the water delivery when the animal is touching the water spout, and then beginning the experiment. So for this experiment, the first time that the animal encountered any odors were actually when we are starting to scan it. So it's completely naive to the task at hand. The only thing that it learns is that if it touches the water spout, a water drop should appear at the end of it. So it comes to this water deprived, so very thirsty and interested in getting the water. So we built a olfaction setup with Professor Dima Rinberg from NYU. It has an 8-channel olfactometer and a non-invasive sniff detection component. You can see here the animal from the side. This is the water spout. It's actually, its nose is inside an odor port. This is what the animal sees. So this is from the front, the odor port, the water port. The nose of the animal is completely enclosed in this odor port. And by looking at the differential of pressure, we can actually monitor the animal exhalation and inhalation cycle. This is how it would look like. An example of a no-go odorant and then a go odorant. You can see that there is even a little increase in the inhalation when the odorant appears, and the animal is leaking multiple times to get the water removed. So this is how the animal will look like for a set of trials after it's already learned the task. So the pinene is the go odorant in this case. It was showing up. Nitrogen is just control of the valve noise when we open the valves. Ethyl acetate is a no-go. The animal doesn't respond to that. Ethyl acetate, again, no-go. You can actually see that the animal is not leaking, but it moved its paws, and then a go odorant. So you can see the behavior of the animal. So how does the behavioral results look like? So one of the motivations to actually go and do our awake imaging of behavior with olfaction as the sensory modality is the saliency of odorants as a driver for behavior. The little clip that I've shown in the beginning of the moving gratings was one of our initial attempts to look at kind of discrimination learning for the visual modality. And in that case, it took the animal about 3 weeks to get to 70% discrimination between the moving gratings. And now I'm going to show you the results of a single animal. This is a pretty good one, but of day after day sessions, daily sessions. So session number 1 was the first day that we brought the animal to the MRI. It's completely naive to odors being presented to it. So this is the first time that within the setup it gets any odorants, definitely pinene and ethyl acetate, which are not something that it's likely to encounter in its cage throughout its life. Every block here is 6 minutes, and each session took place on a different day. So these are consecutive days that we are scanning the animals. You can see here the hits in blue and the false alarms in red. 6 minutes, 12 minutes, 18 minutes, 24 minutes. And what you can appreciate is that almost immediately the animal is at about 70% responses for the odorant showing up. It's actually, the animal is leaking a lot between trials and within trials, so it's not surprising that it catches some of these kind of water drops, or when it's touching the spout, within the trials for the hits, they will get a water reward. But you can immediately see that for this animal, which is a pretty good one, after 6 minutes, or in the second block, it's already showing some learning, so more likely to respond for the hits relative to the false alarms. And by about 15 to 20 minutes, it's already dropping the false alarm responses, going to the 20%, which is its kind of average rate throughout these 3 sessions that I'm showing you. On this last run of the day, you can see that it drops its responses just because it's saturated. So we really have here rapid learning of the animal within 15 minutes, and that again compared to 3 weeks of the first one. This is the response from 10 different animals that are participating in the cohort that I'm presenting now, showing that they are all reaching criteria, most of them reaching criterion either by the second or the third day. We have just this animal that took a little bit longer to learn. So this is a robust kind of a paradigm that all of the animals can learn. You can also see that within a single day, all of them are showing a drop, or most of them are showing a drop in the response latency. So how fast they respond after the order is being presented to them. So again, very robust learning for these animals. So what happens in the brain? So I'll quickly show you a little bit of results for the olfactory bulb and prefrontal cortex, and then some more results for whole brain imaging. What we wanted to do first is, because it was unknown to us what would be the hemodynamic response for this type of stimulation and behavior, we wanted to take an agnostic response where we select regions of interest from the Allen Institute Brain Atlas and just plot the ROIs from these regions. So here I'm showing you 4 regions: the olfactory, main olfactory bulb, piriform cortex, anterolateral motor cortex, and ventral striatum. And you can see robust HRFs. Individual lines here correspond to individual animals for most of these regions across most of the animals. If you now try to look at these responses, you can see already from the HRFs here that we have 2 HRFs. One of them is an earlier one, and one of them is a later one. I'm also plotting as a point of reference the canonical SPM HRF from humans, just so you can appreciate that. And you can see that we have early responses and a little bit later responses. The early responses tend to be more anterior in the olfactory bulb and piriform cortex, and then we see brain-wide responses for the kind of later responses. And you can see the overlap here. This is just distance from bregma as a fraction of voxels measure. So again, these 2 different HRFs capture the responses across the entire brain, and we are going to use that to analyze the GoNoGo behavior. So first, we were interested in looking at whole brain responses to learning. We plotted here 2 different contrasts. One in animals that are already proficient in the task, so already achieved the task criterion of behavior, comparing hit versus false alarms and false alarm versus hits. And you can appreciate that for the hit versus false alarm, we're getting the striatum, we're getting some motor regions responding, again, kind of more robustly because the animal is looking for the reward, making more responses when it gets the reward itself, so just touching, which will also happen in the false alarm, but in the absence of water appearing, that will not evoke water delivery and more responses. And here you can also appreciate that there's some responses for the false alarm in the hippocampus and kind of these temporal regions as well, as opposed to these reward responses. If I'm actually plotting the HRFs, again, for each individual animal, in the thicker line as an average, you can see primary motor cortex, lateral hypothalamus, striatum, granular insula, hippocampus, and just focusing on striatum and hippocampus, now you can see a clear distinction between these 2 regions. It's also interesting to think about this aspect in terms of just reward delivery. So obviously for every time the animal leaked during hit trial, it gets water. So now we have responses in, say, gustatory cortex, a granular insula, from the digestion system, how to tease apart responses that are for the reward versus kind of the learning itself. So here I'm going to focus on the striatum for the hippocampus. It was simple because it shows a flip response. So it actually shows a positive response when the animal is making false alarms. So this is during when the animal is learning, but still when the animal still has enough false alarm responses, so early during this period. So not at the lateral, the end of the learning period. And again, you can actually see that here for the individual animals, they are all showing the same kind of pattern of response of smaller percent signal change for false alarms relative to heats for the striatum and higher for the hippocampus. So deactivation or decreases in percent signal change for heats in this case. So diving deeper into the striatum, this is a region that is highly studied by multiple labs trying to dissociate the different subregions of the striatum in these types of behaviors. Obviously, there are motor behaviors here. There are reward processing, reward processes, reward-related processing processes. And what we are plotting here is responses that are shared between heat and false alarm, so sharing some motor component, and then heat versus false alarm when the animal is performing and looking for the right response. So already in the overlap, so blue for the first, red for the second one, and green, yellow for the green to the third one. And what you can see here is seemingly a dissociation between dorsomedial, dorsolateral, and ventral striatum. We wanted to identify whether we can distinguish between these different regions. Now you see 3 regions of interest that correspond to these different substructures, comparing false alarm and to heat, heat to slow versus fast responses, so motor responses, and finally naive animals versus expert animals, so the learning process. You can appreciate here, again, we see false alarm lower than heats in all of these regions. In this case, slow versus fast. Now, this difference seems to be more for dorsomedial striatum and dorsolateral striatum. And for learning, we see that there's dorsolateral striatum and ventral striatum differences. We quantified that, and indeed we see this kind of distinction between these subregions where correct responses are more in the dorsolateral and ventral striatum. Kind of motor responses, or more likely motor responses, are in the dorsomedial and dorsolateral. And finally, the learning process happens more in the dorsolateral versus ventral striatum. So to summarize this part, and I'm almost done, we see kind of, differential responses in striatal versus hippocampal regions to the heat, but not correct rejection, which I haven't shown in the transition from a learner to an expert. There's preliminary evidence that actual strategies of the animals emerging spontaneously for them are correlated with the rate of learning, and this is consistent with kind of ideas about the contribution of these different regions. Okay, so I'm almost done. I just want to quickly show that we can use this paradigm to also look at reversal learning. So we can now take the same animal, switch the rules on the animal in one of these, after it reached proficiency and it's showing stable behavior. So, and then look at what it's doing. So this is the day of reversal where we switch the animal between the go and the no-go. On 4 animals, you can see here the behavior. So animal reaching proficiency and acquisition. On the day of reversal, these are single runs. And you can see that within that first day, the animal is flipping its behavior, stopping to look for the what used to be the go odorant is now, and now is the no-go odorant. and starting to leak for the other odorants. So again, within a single session, we can look at flexible behavior. Just as a teaser for the regions that are participating, which are highly sought after by multiple researchers in the community, we looked at the region of interest within the orbitofrontal cortex, showing that while during learning it doesn't seem to show a consistent differentiation, it seems to be participating and critically when the animal is actually doing this reversal or figuring out, having its aha moment, contributing to the animals showing flexible behavior. Okay, so I'm just kind of going to put this up. I think that this auto discrimination behavior, I hope I was able to convince you that we can effectively monitor behavior at high resolution. So see the sniffing correlated with behavior, see the licking responses. We see odor and response selective regions. I haven't shown a lot about it, but I've shown you some evidence for that. Definitely showing kind of the engagement of different regions as a function of learning and highlighting that this paradigm can be used to look at systems that might be competing or interacting at different times. Overall, I hope that I was able to convince you that we have here a flexible platform to look at a behaving animal, not just within a single modality, actually in multiple modalities. We already have kind of ideas on kind of combining all of these modalities together to have like a variant of virtual reality for the animal. So it can kind of get multimodal stimulation, engaging in more complex behavior, so linking odorants to different places when the animal is locomoting. And at this point, I would like to also thank the lab members who actually did the work, Eyal Bergman, Daniela Lichtman, Jad Asle, Guy Yona, and Amir Lowen, and kind of point out to the future where we'll combine all of these things, and also just put on the screen as we take questions, the different directions that we are taking. And thank you for your attention.
Thank you very much, Dr. Kahn, for this great talk. And we now move to the Q&A phase. I'd just like to remind the audience to please either raise your hand or type your question in the chat or Q&A tabs. And we have the first question from Iris Chen. Iris, can you please unmute?
Nina?
Yes.
Hold on a second. Can you hear me now?
Yes.
Yeah. So my question, a very nice talk. I really enjoyed it. So my question is that you got this ultra high resolution MRI in order to see those signal changes in the mouse brain. So that's really nice. But I noticed that both the signal changes you presented in the chart is usually like below 1%. So I just wonder is that what's the noise level you typically have within those scans? So with 0.5%, is that, I mean, how much can you overcome with the noise for you to really get the statistical power you need? Was that, do you need to do a lot of like repeating measurement, things like that?
Yeah, so it's a great question. I also see a note about someone saying about the 4% bold changes, and I'll comment on that because these are two related questions. So first, I'll actually mention regarding the 4% bold changes. This we've seen when we done optogenetic drive. And it's a critical difference because in optogenetic drive, we have a lot of neurons that are expressing channel rhodopsin, and it's almost like a hammer hitting the brain. And we are not seeing 4% signal change when we are doing an animal, participating in behavior. So that's an important distinction. I also agree that 4% is a lot to move, and I assign it to the fact that you have so many neurons firing at the same time in a very artificial manner. So that's why I think we saw that response. And again, when the animal is doing behavior, we are not seeing this percent signal change. And now going to the other side of that kind of a lower end, where we get a 0.5% signal change or 1% signal change for some of these behavior. So for some of these trials, we do require 20, 30 repetitions, but I must say that there are some regions in the brain, like the striatum, like the nucleus accumbens, when the animal is doing a lick response or a reach response in motor cortex, that then I think that even 15 or 10 trials are sufficient to get a reliable response. So again, the animal is awake, it's head fixed, so we don't have any suppression of the signals from, say, anesthetics. And then I think that we are getting closer to what you see in humans. There is a question of, of motion. What we've done is, we are actually now also in the process of improving the head fixation that we have. We can kind of throw out trials where the animal is moving too much. It's actually the case that if you hold the animal from its head with this head fixation apparatus, unless the animal is using its paws to pivot, so move, you actually, you might see some movement of the that is subvoxel of the brain, but not a lot. It will actually only move above a subvoxel resolution when the animal is using its paws to push against its head, and then it has leverage to actually create a rotation of the brain. So we don't see a lot of motion, and then in terms of signal to noise, 15, 20 trials are, for some of these kind of conditions are sufficient to get responses. Because we have a lot of data, we are scanning the animal day after day after day. We can, split those and see whether these responses are reliable, that they are reliable across animals. And I haven't shown like individual days and stuff like that, but I've shown you the kind of consistency across animals when you average them together. Again, with spin echo, the amount of smoothing is critical. We usually use something like on the order of twice, almost to 3 times the voxel in plane resolution. So if the voxel is 200 microns, we'll use 400 to 500 microns as a smoothing kernel, and then you get kind of a more spread out response and better SNR.
Yeah, great. Thank you so much. Thank you so much. So is the noise of the magnet an issue or not to the animal?
So it actually, the mice do not react to the scanner kind of noise in the spin echo EPI. We have about 30 something slices, so it's a high pitch very repetitive noise. So the animal is not startled by it. When the scanner starts, it doesn't show like a startle response. So as far as we can tell, and it's behaving. If it would be like very painful noise, it will not behave. If it will be something that annoys it, you'd see a kind of startle response when the scanner starts. So, as far as we can tell, the animal is okay with the scanner noise.
Interesting. Thank you.
Thank you, Itamar. I think you covered most of the questions. Patrick, if you have anything else, feel free to unmute, but I think your question was answered. And I think this is the point where we can thank you again once more for a great talk. And we hope to see you next time in person. You're not that far now.
Yes, thank you. Thanks for having me.
Yeah. Bye, everyone.